Good morning, good afternoon, good evening, everybody. My name is Ashu Tandon. I'm the Chief Commercial Officer at Syngene, and today it is my absolute pleasure to host Professor Ruth Roberts and Dr. Alan Collis for this exciting conversation around toxicology. Ruth is the director and co-founder of ApconiX, an integrated ion channel and toxicology company. She's also chair and director of drug discovery at the University of Birmingham. Ruth is an established science professional bringing rigorous expert thinking to toxicology, drug discovery, and drug development. With over 140 publications in peer-reviewed journals, her work focuses on science-based approach to drug discovery and development, especially in reducing attrition that's attributable to safety and toxicity. Ruth was also the recipient of the SOT Achievement Award in 2002, the Eurotox Bo Hempstead Award in 2009, and the SOT Founders Award in 2018 for outstanding leadership in toxicology. Prior to this, Ruth was Global Head of Regulatory Safety at AstraZeneca between 2004 and 2014, and Director of Toxicology for Aventis 2002 and 2004. Ruth, I always say that in pharma, we all age ourselves by the companies we used to work with. I have very fond memories of Aventis before the acquisition by Sanofi Synthelago. So great to have you on board. Welcome. Thank you so much.
Thank you.
Yeah. Alan Collis has recently joined us as the Vice President for Integrated Drug Discovery Services. Alan is a drug discovery and development scientist specializing in discovery, preclinical, and early clinical project leadership. Alan has served the European and US markets in various leadership roles with responsibilities including medicinal chemistry, DMPK, safety assessment, CMC, and phase 1 studies. Alan has led 33 small molecules into preclinical evaluation by partnering with a diverse set of teams, both internal and external, and has initiated multiple clinical studies of new science. His prior experience included working with Pharma Therapeutics and Novartis. Prior to joining Syngene, what was it, about 7 weeks ago, I think, Alan? Alan was the Senior Vice President for Preclinical Development at Nimbus Therapeutics. And earlier on in his career, Alan has had significant leadership roles in medicinal chemistry in large pharma companies such as Aventis, Roux-en-Poulenc, and Pfizer as well. Welcome on board, Alan, and thank you for joining us.
Thank you so much for the introduction.
Great. So, one of the first topics that we're going to touch upon are what are some of the key trends that we are seeing in the toxicology space. I know there's a lot of action happening over here. And I'm going to start with you, Ruth, with the first question. What are some of the key drivers and challenges that you see in the market currently? And how do you think they're expected to impact the market? And, both from a drug discovery pharma company standpoint, and if you could also cover the angle from a service provider perspective as well. And I think they'll be sort of interlinked as well.
Yeah, thanks, Ashu. That's a great question, and I think one that many of us have thought a lot about over the years. And in response to how are things changing, what current trends, if you like, in the toxicology market, I think I'd divide it into three topics, three ends, if you like. The first thing that I think is really impacting what we do in terms of science and also delivery of services is the new modalities that we're seeing so much around in the field. I recall when antibodies were a big deal over new molecules, and then it was bispecific antibodies and various types of biologics. But now, with oligonucleotides and inhibitory RNAs, targeted protein degradation therapies, there's a constant evolution of new modalities that is really exciting, but it is challenging as the scientists to think about how we move these quickly and efficiently but safely towards the clinic. So that's my first, is N for new modalities. My second would be N for non-animal methods. And I think that will come up again later in our discussion, that we are constantly thinking about how to do things differently, to approach the science differently, again with that common aim of rapid and efficient movement from the bench into patients, but whilst ensuring we protect volunteer and patient safety. So that obviously is a science-led initiative, but then it also impacts on how we provide services and how we plan and deliver the toxicology package that's required. The third thing that I think is something I spend a lot of time thinking about, because I'm director and co-founder of ApconiX, but I have also, I'm also director and chair of drug discovery at the University of Birmingham in the UK. So I spend some of my time in the academic environment. And what's really apparent even more over the last 5 or 10 years is when I joined Big Pharma, the innovation was taking place in Big Pharma, whether it was Zeneca, Sanofi, Aventis, AstraZeneca, Pfizer, GSK. Increasingly, we see a lot of the innovation taking place in academia and in small biotech. So N for novelty is the novelty is often now coming from biotech and academia, which is excellent because it means that you see such diversity in approaches, but equally well often people, the scientists in these companies are not constrained, and I use that word advisedly, by tradition of what has been done before, and are seeing a toxicology package design as a blank piece of paper, which personally I would love, because that's how I think as well. Let's understand the science program, let's think about what's right for this, and then deliver it. So that would be my thoughts on what's new and evolving trends in the toxicology space would be new modalities, non-animal methods, and novelty in approaches.
Thanks so much, Ruth, for that. Alan, do you have a point?
Yeah, I just wanted to add a couple of comments. In my experience, the first half of my career was all with Big Pharma, and particularly in safety, it's gone from an institutional model to a distributed model. When I was first in Big Pharma, it was like, we will never, ever outsource our GLP tox, and certainly not our registration tox. Right? It's never going to happen. It's never, right? Yeah. And now you look at it, and it is in a much more distributed model. And I like Ruth's point on, fresh minds and fresh thinking. There's also an absence of institutional expertise sometimes, having consulted with some of the biotechs as they're going forward into evaluation, just to help them understand what's required. And even that finding safety in toxicology signals is a really good thing, particularly if it's reversible and monitorable. You really have to have that to actually carry the thing forward, So, yeah, I think it's changed a lot, and there's a consolidation on the providers as well. If I look, 8 to 10 years ago, the number of CROs or providers I could go to do preclinical safety was a much longer list than it is today. That part of the industry has also changed quite a lot.
Yeah, I think, Alan, to your point around lack of institutional knowledge with companies today, I see a lot of people following the same path that you have, of having worked on the buy side or on the scientific side with a big pharma or a small biotech company for a number of years, and then now moving on to the CRO, CDMO market. So there's a lot of talent that's obviously sort of now flowed into that market, which does end up giving customers the great sense of confidence in the ability to get things done according to standards and protocol, as the case might be. So I think that's also been a key part of the evolution that has helped in this space. Yeah, great. So Alan, one of the questions that I have for you in a similar topic is, what are some of the new ideas that you're seeing from a non-animal approach standpoint to predict repeated dose systemic toxicity?
I think I'd probably let Ruth lead off on that one. I think she's closer into the centre there. So I don't know if you want to just offer a few comments first, Ruth, and I'll pick it up from there.
Yeah, absolutely. So, I have been heavily involved with this over the years, Alan and Ashu. I'm passionate about non-animal methods and doing things better and differently. And I know certainly in the UK and in Europe, the 3R, reducing, refining and replacing animals, is a big deal. But it's not just about that. It's about using that as a driver to do better science and to use more predictive models as we move into patients. So there's a lot of really exciting things happening in this space. And the thing I particularly like to draw people's attention to, if they're not aware of it, is a project that I've been involved with over the last 5 years with the NC3Rs, the National Centre for the 3Rs, which is based in London but works globally. And we started a project around 5 or so years ago where we were asking the question, based on guidance on biologics under ICHS 6, do we actually need 2 species for small molecules, or could we learn from sometimes biotech programs successfully get into the clinic with 1 species? So the question we set about asking was, what is the value of that second species in defining the toxicology profile of a new molecule? And we engaged with maybe 30 CROs and pharma companies worldwide and asked them to submit their case studies. And we had 130 or so case studies of what was seen in the first species, usually the rodent, and the second species, dog, non-human primate or mini pig, and whether we learned anything from that second species. So was it needed? Now that piece of work is published, and I'll be very happy to share that with anybody who's interested. So Helen Pryor is the lead author. So we concluded that we can replace the second species for certain molecules. But what's now out now is the NC3Rs have a thing called the Crack It Challenge, where they put the challenge out there and companies can submit potential solutions. And the Crack It Challenge is called Virtual Second Species. So we're looking for ideas now on what could be that virtual second species, and it's focused very much at the moment on the dog, just to more closely define the challenge, because it could be too big otherwise. But I'm on the panel of what's going to be coming forward to that, so I'm really excited to see. But I'm expecting to see everything from machine learning through to the organ-on-a-chip methodologies. And then hopefully I think the theme of them will be human-based systems, whether it be modelling human data or organ-on-a-chip microphysiological systems based on human tissues. So I think we're really on the edge of some game-changing technologies here that will change how we do this.
Wonderful. Alan, any comments?
Yeah, I was just going to add, there's a strange, almost conservative force from the smaller biotech perspective as well, in that they don't want to get novel and creative with the FDA and the IND submission. They want to do something that's proven and certain, and so I see both dynamics in play. But I think the 3R initiative is very important, and it is such a heavy investment when we go into both species, even for the IND stage. I think to keep looking forward for that. But I think cross-learning, we need to build on experience as to how well and how effectively the new methods will supplement what's required from a regulatory perspective. I've never got daring enough to ask them to try and go beyond 2 species, to be honest, Ruth. I mean, maybe I'm too conservative.
That's one of the topics, Alan, isn't it? It's who is more conservative, small biotech, big pharma, or the regulators? That is a long discussion. I think we could have.
It's not a simple discussion, surprisingly enough. You would think that the biotechs are tremendously, but they're not actually. They tend to be very conservative.
That's great, yeah. No, I think that's good. We do have some questions coming in, so as I see the relevance, Ruth and Alan, I'll pass the norm. But there is a question from Nick Tezapsidis, who's asked us as to how can developers.
Starting point was, do we actually already have the data we need to support human safety based on modelling? And then what would you absolutely have to do to fill in any gaps or do bridging work? But again, we've seen lots of repurposing where you make a really good scientific argument to the FDA or the MHRA or the EMA saying, actually, for this repurposing indication, we know what we need to know and we don't feel any extra work is needed. Does the agency agree? It would depend on what you're repurposing for. If you're going from oncology to asthma, then probably that could be quite challenging because the safety package might not support it. If you're changing dose routes, that could be challenging. But largely, I would start with what are you trying to do? What safety questions does it raise and what data do you already have to support that? And then talk to your agency, because there's fabulous expertise within the regulatory agency.
Yeah, I would just reinforce that point at the end. When I think of that question, I think of the pre-IND meeting. I think you'd want to set something up. If you want to do something a little bit unusual or streamlined, great, let's do it, because I think that's helps change and transform our practices. But do it in discussion with the agency. You set up some time with them and you can at least get a written response as to whether they're on board with your approach or not. And they're open there. I don't think of the agency as a police force trying to beat you up or find fault. They're actually looking to partner, and they're really interested in listening to well thought through science. I actually don't think that the regulatory agency necessarily is the primary conservative driver on preclinical safety approaches.
Yeah. No.
There was a related question which I loved actually, which was from John. How can we ensure that promising candidates are not rejected incorrectly at the toxicology stage? I comment very briefly on that. It's sort of like a related question, but it's hard because it's not a testable hypothesis. Once you've killed a compound, you don't tend to do much more on it. I think that I would say that make sure that there's very good debate on the data to prove that and gain confidence that it's compound related. What's the confidence of the translation through into a clinical setting? Get a group of experts together, right? Before you take a final decision to pull something. Doing wise preclinical safety, it's not a ratio game of, my answer is 7.3. It's the nature and the monitorability of the toxicities and safety signals and how they'll translate into the clinical trial setting. Ruth, what would you like to add to that? It's a very common thing that I'm sure you've wrestled with. How dare you kill my compound?
There is. I'm amused by that question. Incorrect rejection, because if you set it's about specificity and sensitivity, isn't it? If we set our systems too high, we throw out the baby with the bathwater, and if we set them too low, then we are allowing potentially toxic molecules through. So there is no answer to that. We will always be potentially rejecting promising candidates, John. However, the point is that you should have many candidates that you're considering at the LO phase, so that you're selecting the best of 10, say, the best few from maybe 10 LO molecules, so that what you're taking forward has got the best chance of succeeding. There is no answer to that. We will inevitably throw out some that could potentially be successful. It's just a numbers game, isn't it?
Yeah, and I think one of the changes you have seen is several companies like to take 2 or even 3 compounds into preclinical dose range find, at least in the rodent, to see if there's differentiation between close analogues and then pick the best one, rather than go for the single compound sort of Hail Mary pass approach in the hope that it gets through.
Yeah, but if you imagine you're in a project team and you're, it's just that question, Alan, how dare you reject my molecule? Is, we need diversity in our hit to lead and lead optimization, so that we can choose the candidate that's most likely to be successful, all things considered. There's some great questions here. I just want to pick up FRAMs. When do you think reg authorities will accept the replacement of second species? I've only picked on that one because I don't think that's the way to think about it. I think this is, as Alan said before, we are a team with the regulatory authorities using our scientific collective know-how to make the right decisions. So it's not us suggesting something and the agency saying yes or no. It's a collective effort.
Yeah, and I would point to a couple of illustrations of I've seen changes in practice over the past 5 or 8 years. If you look, for example, on genotoxic impurity analysis, in silico approaches for that are pretty well accepted now. From a DMPK perspective, the need to do clinical DDI studies, the FDA and the other regulatory agencies will listen a lot to, the sophisticated modeling approaches, the SimSet and the physiology-based approaches, to say whether or not you need to do that clinical trial. And if those expert approaches are saying don't do the trial, the FDA will say you don't need to do the trial, right? So I think there are traction points in there. I think with safety and toxic signals, it's more complex. It's harder to get tractable, but there is progress, and I'm sure it will continue.
Yeah, no, that's great. So maybe this is a good segue into sort of talking about emerging therapeutic modalities like targeted protein degradation or cell and gene therapy. Are there any specific toxicology challenges that you see for some of these newer emerging modalities or platforms?
Yeah, let me answer that from a project leader perspective, and then I'll pass it over to Ruth, I think, through for comment. One of the assumptions that many people make on safety is they go for a trough level of concentration of your compound relative to on-target or off-target activity, and they think that that's the truth and that's the answer. These new modalities, such as the PROTACs, from a exposure effect point of view, a PK/PD point of view, they're not direct response. So you dose, you get a degradation in protein which lasts for a certain period of time. How do you then select when do you dose again, right? If your dosing is just based on a PK trough level relative to an IC50, it's a very straightforward calculation. It's like antibiotic field, it's like a deeper histories. It's harder in this area to know how frequently you should be dosing. And when how frequently when you're dosing preclinically, how are you going to translate that into human? My concern a little bit, having spoken to people working in this area, is with a classic pharmacology, a classic preclinical safety approach of once daily dosing to an MTD and then titrating down, that might not be a clinical modality. Now, it's easy in the oncology space to tweak those things around. They're used to doing that, and the clinical trial support is flexible enough to allow you to do that. I think it's more complicated if you get outside of the oncology area, because once you've defined a safety exposure limit, that's going to sit as a ceiling on your clinical strategy. But so that's a project team leader's perspective. To a toxicologist, what do you think about it, Ruth?
Not that we're on opposite sides of the table, Alan. We spend together. I was going to comment on this. I had a really interesting experience last week. So I work with HESI, the not-for-profit organisation based in Washington, D.C., that exists to bring regulators, scientists and pharma together to tackle difficult scientific issues. And last week we had a workshop on targeted protein degradation as a new modality. And it was so interesting. I was co-chairing it with Lynn Jones from Harvard, and I went into it thinking, the way we set the workshop up, we had folks from lots of small, medium, large pharma with case studies. The starting point was, how do we design and deliver toxicology packages for PROTACs, targeted protein degradation therapies? And it's already gone beyond oncology. It's already some inflammation projects. So we're out of the ICH oncology guidelines and into M3, which requires, slightly more stringency. And it was absolutely fascinating. We had a few days last week on this, where collectively we narrowed down the challenges to be the obvious ones for a toxicologist, which is choice of species. How do you address safety for targeted protein degradation, which species are right? And for those who don't know, so targeted protein degradation is also one of the strategies is a molecular glue, and molecular glue is what thalidomide was. So there's big issues and lots of questions. The questions that came up were around early de-risking. So how do you understand on and off target with protein degradation? So at the early stage, and there were some people using cell lines, some people doing proteomics. It was really diverse. How do you address early ADME PK/PD for these molecules? And they do things like absorb all the endogenous ligases, so you then get unusual findings. And then what kind of early de-risking, and then what choice of species? And we ended up deciding that what was really needed is to set out a framework for decision making in this space. But it was so fascinating. I went into this workshop thinking, is this going to be like a small molecule or an antibody? And I came out thinking let's just start again, really.
It's something else, yeah. Was there much debate on the bell-shaped dose response or the hook effect that you see as you push these things up? Because that's a complicating feature as well. If your pharmacology is coming in and going away as you're doing your dose escalation, I'm from a more classic sense, I'm scratching my head about how to interpret margin when you've got that.
Yeah, that was a challenge, how the framework by which you would do that. But the choice of species was the real discussion, because there are different expressions of different ligases in different species. And if these targeted rely on engaging endogenous ligases, then that then changes the safety profile. So I think we need to really think back to basics with this and understand what we're doing molecularly, and then design a strategy rather than looking at what we've done before.
So in terms of sort of looking at some of the newer ways of sort of working, what role do you think things like artificial intelligence would have in sort of supporting an overarching tox strategy? Has this really come to, is AI really come to real terms in terms of contributing to building an accurate tox profile for drug candidates, or is it still sort of in the lab, so to speak?
Yeah, I think that's to you, Ruth.
Ruth, yeah, that's
Well, yes. Around, it was super interesting. But what I contributed to that is the bit of this that I understand, which is how we are using artificial intelligence and machine learning in early de-risking of targets, because that's what we do in my company. We work closely with using AI to make sure we assemble the right data sets to then do the very important task of interpreting all of the data to fully understand the unintended consequences of inhibiting a target, then using that for a risk mitigation plan. So that was really interesting. But then what I learned in this workshop is that the FDA has got two very important work streams at the moment. It has an action plan called SAND. I don't know if you're aware of this, which is artificial intelligence and machine learning based software as a medical device. It's called SAND. So it's really worth going to take a look at that, because the protagonists in the workshop are arguing that this is direct evidence that regulatory science is ready for AI. And by regulatory science, I mean the package and the program that we put together. The other was iSTAND, which is innovative science in R&D. And that's another FDA initiative that's talking about using machine learning in designing, in interpreting the package. So both of those, and I can send those to you later so they could go out to the chat. So I think it is really interesting listening to all this discussion, because there's, at the one hand, there's the very early use of AI machine learning around, data analysis, collation and interpretation. And way at the other end, the FDA is thinking about how it's used in regulatory decision making. But the bit in the middle, I'm not sure I quite really could really answer about the bit in the middle around, interpreting data from GLP studies and so on. I don't think we're there yet with that.
Yeah. Yeah, and again, I would just add, Ruth, that I think that, we end up with pretty large data sets. So using more interrogation tools, machine learning, artificial intelligence, or there's a whole suite of other things you can look at, I think it's very valuable. A challenge is, how do you integrate together? Because if you think about it, you've got this aspect of systems biology, which sits back a little bit for target selection. You've got aspects of off-target activity versus on-target toxicities, which is something you can factor in. There's then the issue of exposure, and how does the exposure look like, and how does distribution, even of a particular compound, because that can also impact the safety profile in quite a strong way. I guess more in a multi-year vision, it's how do all of those things get integrated through together. We do have very large data sets. I think one of the questions sometimes that comes up is it the right data, right? You do a phase 3 clinical trial, you'll have oceans of Excel spreadsheets with information on lots and lots and lots and lots of patients. But it's a bit like a proteomic analysis or a gene expression analysis. You've got hundreds and thousands of signals. How do you make sense of it? I think ultimately the answer would have to be, how do we make sense of it in a reliable way that speaks to clinical safety? So it's a good aspiration to have. I'm not sure that it's going to be resolved in 2022. As you say, there are initiatives underway. So.
It's the age-old problem, Alan, isn't it? Is because we've got a little bit carried away with the technology, we maybe haven't thought about what the question is.
Yeah.
If we can closely define the question, then I think AI is proving extremely useful. And I'd encourage anyone to go and look at the 2 FDA initiatives that are, one's an action plan and one's a work in progress. But the other thought I would say, building on the repurposing question we had earlier, is where I've been working with a team at the FDA where I've seen AI machine learning being really useful, and we have published on this, is in repurposing.
Yeah.
Because if you think about it, what you're trying to do is match a whole bank of very complex drug profiles with a whole bank of complex human genetics, looking for an inverse relationship between the genes that this drug down or upregulates and the genes that are down or upregulated in a disease. And you can use that to train the model to then ultimately start to find matches, which I think is a really clever application. But then obviously you would then need to validate that. But I think that's really interesting.
Yeah. I'd just like to sort of move on to a question a topic that we had touched upon in the introduction, right? When we talked about sort of how the trends and how partnerships are evolving. But when you look at outsourcing partners like ApconiX or Syngene, and I want to talk a little bit about the role that these partners have in sort of creating the right toxicology strategy. So maybe, Alan, I'll start with you. What are your tips for drug sponsors when they're going out looking for a toxicology partner? What do you ask them to take a look at?
Yeah, I well remember doing this for the first time because I came from a medchem background, and I was walking blindly into doing preclinical safety evaluation. So I did at least pause and ask a few people which were much more experienced, and the feedback was really interesting because they said, it's the quality of the conversation that you get. I mean, the first thing you always look at is the technical capability. Are they GLP compliant? Will they expect all of those things? And the partners are pretty well placed from that. But having lived this through a few times as well, I think the quality of your partnership with the study director on that particular study is a key success factor. And that's a bit of a social question, right? So you can reach out and a company can tell you what it can do. But then you need to speak to the assigned study director and figure out, are they experienced? Is the interpersonal chemistry right? Because trust me, there's going to be a lot of conversation when you start going into the studies, both about coordinating the events going in, adjusting the study design, interpreting the data, dealing with conflicting views on the data. So that engagement, I think, is crucial. As a very empiricist sort of person at the moment and looking at today, I would also say there's such a long lead time. The demand on relatively few CROs. I tried to place some studies earlier in this year, and it's like, yeah, we can do that, yeah, and we've got the right people, but we can't start it for another 9 months, right? Which for that small company wasn't an acceptable answer. Right? We can't sit and wait for 9 months and pretend that we have some other part of the portfolio to work on. So I think the accessibility to getting studies started, and the other big experimental pressure for us right at the moment in the safety zone is on non-human primate provision and just getting those studies underway. That's a headache wherever you go in the globe, I think. Ruth, how would you build out on that?
Well, yeah, I think I would take the question back to what Ashu asked. It was around outsourcing and what you'll be looking for. So outsourcing to me, because I'm working at ApconiX, it means two different things. So people outsource to us at ApconiX. In terms of lab work, it's for SIPR and anything ion channel, which is what we do in our lab. But the main part of the other big part of our business is outsourcing to look at target safety assessments, and also outsourcing to access toxicology expertise in terms of project expertise, and also, the diversity of expertise that you might need, ADME, DMPK, reprotox, genetic tox, all of that. So in terms of outsourcing for animal work, I completely agree, Alan. We then outsource from ApconiX to other CROs, and what we're absolutely looking for is that relationship with the study director. And on top of that, top quality communication in the moment, I think, is vital. You need to know what's happening in your study at any time. But when it comes to the work that is outsourced to ApconiX, target safety assessments and project toxicology, we're seeing, when we started ApconiX 5 or 6 years ago, we thought our primary clients would be small biotech that didn't have the toxicology expertise that we have, and we could help design their non-clinical package and help them through. And indeed, we do have many clients like that, but it's grown massively to be medium and large pharma, government and academia as well. And I'm learning myself that the reasons why people outsource to us is exactly what you'd expect: resourcing to peak, so they don't have the people to cope with the volume of work, whether it's target safety assessments or project work. But then we're also seeing a lot of outsourcing to access the required expertise, because they might not have that in the company. And that links into what we were talking about earlier around innovation, where, as companies are moving into new modalities or novel spaces that they might not have the expertise in-house, they're reaching out to collaborators across the globe to help with that. So I think people are outsourcing for access expertise to help them through peaks. And then another trend I've seen more recently is, particularly within large pharma, the expert toxicologists that they have in-house, they want to keep them project facing. So they're not spending their time doing things that could be outsourced. So anything that can be outsourced is being outsourced, whether it's target safety assessments or indeed whole IND packages, so that the internal experts can be in those project team meetings, building relationships with the biologists and the project leaders and so on. So it's changing all the time, and it's just really interesting to watch these trends.
You mentioned a couple of pointers, and I think the whole aspect about focusing on sort of what requires maybe internal interaction, interfacing, more strategic activities versus sort of the more tactical stuff that can be outsourced is sort of one of the areas where people can look at. Are there any other pointers that you would give to us in terms of how you see toxicology outsourcing evolving over the next few years?
Yeah, I mean, there's several trends here, Ashu, that I've seen. I'm with Alan on this. When I first started in this space, working in drug discovery and development toxicology, outsourcing GLP toxicology studies was absolutely out of the question. They were we started by outsourcing carc studies, then the chronic studies, but the GLP were absolutely regarded as being in-house. Then they went out in big pharma, and now we're seeing the dose range finding sometimes done in-house, but then that's a bit of an awkward break to do the DRF in-house and send the GLP out, because you might be we all know it doesn't always necessarily translate. I don't understand why that is if you've got the same strain, but anyway. So what I'm seeing is, increasing in outsourcing in this market. And I think trends to look out for in this are as drug discovery and development is changing, new modalities are changing, then we're seeing increasing challenges on how we deliver these studies and how we interpret them. I think that's going to be really key to watch, and seeing essentially people outsourcing whole tranches of work and things that were to me unthinkable, like the whole IND, just put a whole project out. Usually backup or low priority projects, to be honest, but just putting an entire package out to a CRO and saying, please just run with this all the way from, lead to LO through to IND. That to me,
as well as multiple cities in India as well attending. So it's a really great group of people that I think we've got over here. Manoj Jadhav from New Jersey asks that it's a very specific question, that how should one go for tox studies for a repurposed for oncology indication? It's a combination of two drugs, one of which is approved, the other is at an IND stage, and tox in dogs is completed. Any suggestions? I know it's a very specific one, but do you have any answers for Manoj?
Yeah, again, this is an area which has changed in oncology. There used to be a time when you could, the oncology division is much more open to putting two experimental drugs together in a combination study. But you need to design the study in a way which allows you to retitrate a little bit on the human tolerability. So how much do you need to repeat preclinically? A debate I've seen often come up is where the primary indication or the primary clinical use is going to be with another agent, right? So it's always going to be partnered, with mechanism Y. Should we be doing the toxicology just in with mechanism Y? How much do you want to do on your registration toxicology preclinically on looking at the combination work? But I actually think the environment's relaxed back a lot on this question over the past 5 to 8 years. There was a time when, it just wasn't allowed. You couldn't take 2 experimental drugs and put them in side by side. But I think the FDA will listen to argument, and maybe it's a little bit down to that pre-IND type discussion as well for the FDA. Ruth?
Yeah, I absolutely agree, Alan. And I think what we're seeing a lot in the chat generally is a lot of very specific questions that are quite focused on what I'm reading as being specific challenges that people in the questions are facing. And I think this illustrates the point that both Alan and I are making, is that it is not one size fits all. Every project is different. Therefore, we can't really answer these questions because we'd need to know a lot more about it. We need to sit down and find out what you already know, what combination you're talking about, what territory you're in, and what data you have already, and then we could answer the question. But I do agree with Alan that the agencies are much more open to innovative approaches in this space. So I always design something that makes sense scientifically and then speak to your agency. In the UK, the MHRA now has an innovation passport, which is a new initiative where once you've got an innovation passport for doing something new and different, then you're kind of fast tracked. So that's worth guys having a look at as well.
That's really interesting. We probably have time for just one more question. So Juan has asked us, for clinical trials, is it possible to submit a more diverse side product candidate and have those toxicology results be relevant to the same active with fewer of those side products?
Again, I can't answer that without knowing what you mean.
Yeah.
I think what you're kind of saying is, can you submit one molecule and then have those results be regarded as relevant to a similar molecule? That's what I think I read there. But again, it would depend what does side product mean? How similar is it? Why are you? And more importantly, what are you trying to do here? It could help. It's kind of hard to answer this question without knowing exactly what you mean.
I was talking a little bit about openness from the FDA. This is one of those areas which is rather less open, I think. They'll look at the CAS number and the route of administration, and if everything matches up, they'll say that proves something. If it doesn't, then you really have to reprove it at the moment.
And my question, why are you doing this?
Yeah.
If it's the same, then of course you can. If it's different, but then if it's the same, what's the point in doing a diverse side product? Because you're going to get the same results in the non-clinical and the clinical. But I take it's diverse enough for you to expect different results. Therefore, you cannot extrapolate one from the other. It's just illogical. It doesn't make sense scientifically.
Yeah.
No, I understand. Well, I think thank you so much, Ruth and Alan, for attending today. I wish you the best, and thank you for the work that you both do.
Yeah, thank you for the questions.
Thank you.
Thanks, Ruth. It was good to speak.
Right. Thank you.